I'm currently a PhD student at the University of Utah in the Department of Medicinal Chemistry. I graduated from Westminster with a bachelor's in neuroscience and chemistry in 2018. We're here today with Jamie Butler and the Great Salt Lake Institute at the Great Salt Lake Summit that's hosted by speaker Brad Wilson, and we are sharing some of our research, some of the things we do at the lake to help advocate for water policy and spread the word about all the interesting things that are happening at Great Salt Lake and impactful research we're doing there. I had some really impactful experiences with Great Salt Lake Institute and the research they supported while I was at Westminster. My research was mostly with Dr. Frank Black studying trace element cycling in the Great Salt Lake. All of the research opportunities I had during the summer and the school year, and faculty really pushing us to explore our own ideas and supporting us publishing while I was an undergraduate, it really helped me get excited about science and want to pursue a PhD. I'm now working on a PhD at the U. I'm in the medicinal chemistry department in a natural products drug discovery lab led by Dr. Jacqueline Winter. And my project is involved in looking for new antimicrobial agents that are produced by Great Salt Lake microbes. This summer, Great Salt Lake reached a historic low water level. And if that trend continues in the following years, we could have some really terrible catastrophes at the lake. This could impact the microorganisms, all of the birds, a lot of the industry surrounding the lake. With a shrinking lake water level, we're going to see a lot more dust generation in the valley too, so that could impact human health across the Wasatch Front. Overall, we've had a lot of positive feedback and optimism from lawmakers. It seems like a lot of people have the same motivations to protect this lake and this ecosystem. I hope that they just get more examples of how important the lake is to all Utahns, whether it's their jobs or just their health. Follow Great Salt Lake Institute for more lake knowledge.
Hi, everybody. Welcome to another episode of Salty Science. Today, we're coming here on March 15th of 2022, and we are here to talk with some really amazing people. Before we start, I want to remind you of some activities that are going on. Again, my name is Jamie Butler. If you don't know me, I am the coordinator of Great Salt Lake Institute at Westminster College, where we work to connect people to Great Salt Lake through research and education. I'm really excited to be coming here after this legislative session was so successful, and I hope that you will reach out and thank our legislators for doing a lot to support Great Salt Lake through the legislative process. Despite the fact that they did not pass the brine shrimp as our state crustacean bill, I want you to know that it didn't fail. They didn't fail this bill and kick it out. They just didn't have time to deal with it. And so hopefully we will see that next year. Today we're here with Dr. Jackie Winter. I brought you first Abby Scott. Abby worked with Great Salt Lake Institute and was actually one of the undergraduate researchers that wrote one of the chapters in the Great Salt Lake biology book, Terminal Lake in a Time of Change. And after leaving Westminster, she went to Dr. Jackie Winter's lab. So we're really excited to have her there. All of these webinars and all of the things that we've been doing can be found on our YouTube channel that you can access through this QR code. And we're kind of going on to fieldwork season. We're getting busy starting to prepare for fieldwork, so we probably won't be joining you virtually for a while. But we do have the Great Salt Lake Bird Festival coming up. Today at 9 a.m. you could start registering for trips. A lot of those have already filled up, but I did notice there were some beginning birding trips, and there were some really cool trips to take that are May 12th through the 15th. And then you should come to Great Salt Lake Issues Forum. This is the premier conference put on by Friends of Great Salt Lake that will be May 18th to 20th, and will be all things Great Salt Lake that you can find on their website. And so I want to bring you Dr. Jackie Winter. Come and join us, Dr. Winter.
Hello.
Hi. And
Hi.
This one's really, I never thought I would be talking about the antimicrobial properties of Great Salt Lake microbes. And so I'm really glad to have Dr. Jackie Winter here from the University of Utah to talk about the research that her and her really super cool lab are doing. So join us, come tell us about your microbes, Jackie.
Thank you so much, Jamie. And thank you for this opportunity. I mean, we are so excited about, using the Great Salt Lake as a resource, but I think more importantly, as, you pointed out, and as Abby, in her beautiful video, we all want to be advocates for the lake and preserve this precious resource. And so anything we can do to help save Great Salt Lake, count us in, sign us up, we'll be the advocates. And so, today I'm going to be talking a little bit about some of these microorganisms, and as Jamie just mentioned, their utility for producing therapeutic agents. And primarily what we're using these microorganisms for is for the discovery and development of new antimicrobial agents. And I always like showing this photo of the University of Utah here nestled in on the Wasatch Front, because about 15,000 years ago, Great Salt Lake, we could think of as the Salt Lake Valley, would have been underwater with prehistoric Lake Bonneville, which at the time was a freshwater lake. And I believe it was about the size of Lake Michigan, so a huge, huge lake. And then when that drained about 15,000 years ago, Great Salt Lake is what remains. And so let me see if I can turn off and progress with slides. All right, so normally when you think about microorganisms, and let me hide my, there we go. When you think about microorganisms, the first thing that normally comes to mind are disease, crop disease, human disease, food contamination, but not all microorganisms are trying to kill us. They can be used for good. And if we look at this, we can, again, my slides want to advance. Microorganisms can be used for fermentation of, foods and beverages such as sake and blue cheese, but they can also supply us with therapeutic agents. Some are shown here, such as lovastatin, penicillin, doxorubicin, amphotericin B. And it is these therapeutic agents or these natural products that my lab is really interested in. And so these natural products are small molecules, and all kingdoms of life produce natural products. And we can classify natural products or small molecules as essential. We could think of these as amino acids, as carbohydrates, as lipids, or we can classify natural products as sort of specialized compounds. Not all organisms are going to make these compounds. And organisms that make these specialized compounds may be doing so to enhance their fitness, or maybe to adapt to a certain environmental niche. Some of these natural products have great therapeutic utility, and some are just on this slide, such as the anti-cancer agent paclitaxel, doxorubicin, anti-parasitic agent ivermectin, antifungal agent echinocandin B, antimicrobial agents vancomycin and tetracycline, and then immunosuppressant agent cyclosporin A. And we know that these natural products have great utility. And I think if you see any natural product talk, you'll normally see some kind of pie graph to show that these small molecules are still important for drug discovery pipelines. And so this is a review put out by Newman and Craig about 2 years ago, and kind of summarized over a 38-year timeframe how these natural products really fit into the drug discovery pipeline, where these small molecules are either themselves used as therapeutic agents, such as antibacterial agents, anti-cancer agent, or other therapeutic applications, or they're inspired. Their pharmacophore inspires these small molecule drugs. And we can envision that, nature is making these compounds. They've evolved over millions of years to interact with specific biological targets. And so maybe we use some of those pharmacophores that are acting or, hitting those targets, and then can modify those agents. And so natural products are instrumental in the drug discovery pipeline. And so if we think about, using these as an inspiration, again, we kind of go back to where we can use those pharmacophores. We can use those to make more potent agents. Maybe we can increase stability, decrease toxicity as observed with tetracycline and the newer generation minocycline. We can even overcome antimicrobial resistance, such as the development of tigecycline with this T-butyl aminoglycyl amidyl group. And even with lovastatin. So for those who are taking the anti-cholesterol agent lovastatin, this is a natural product. It's produced by Aspergillus terreus. And if we just swap out, or I guess change this diketide side chain from containing one methyl group to this gem dimethyl is observed in simvastatin, we increase the binding affinity to that hydroxymethyl-glutyl-CoA reductase. And so this is really kind of highlighting, again, natural products and their involvement in drug discovery. But a problem, though, with using natural products, and in the previous slide, just showing this, the chemical diversity, is that some of these chemical modifications can be very difficult to replicate using synthetic methods. And this can be problematic when moving this material through downstream applications, for assays, or even getting tangible yield for actual drug development. However, we can turn back to nature, not only for the chemical side, but looking at the biosynthetic machinery. Because with nature, we can look at the blueprint and look at the genes that encode the biosynthetic machinery that makes these compounds, and use that as a guide to sort of figure out, how does nature make these compounds? Because in bacteria and fungi and in some plants and even in some animals, the genes that encode the biosynthetic machinery are usually clustered together in the chromosome, and they make these biosynthetic clusters. And I have an example shown here. The biosynthetic cluster involved in the biosynthesis of lovastatin, again, that anti-cholesterol lowering agent produced by the filamentous fungus Aspergillus terreus. So what these arrows represent are open reading frames, and they point in the direction of transcription, and they're going to be color-coded based on putative function. So we'll see a lot of these biosynthetic clusters as we go through this lecture. And what we can see is that nature can take very simple building blocks, such as malonyl-CoA, and through an assembly line-like process can build these beautiful, diverse structures containing multiple rings, multiple stereocenters, functional groups. And we can kind of, when you start thinking from a synthetic chemistry perspective, can start envisioning how difficult it could be to make these in the lab. But what my lab can do, and a lot of other labs in natural products, is we can look at the blueprint that nature uses and say, okay, how does nature make these compounds? And can we use these enzymes as biocatalysts to replace some of these synthetic procedures in actually making these compounds for downstream applications? And so in my lab, we kind of call ourselves the jack of all trades, master of none, and that we use a variety of approaches for developing and identifying unnatural products for therapeutic applications. We can go out and we can isolate bacteria or fungi from environmental samples. We can ferment these in the lab under a variety of culturing conditions, isolate bioactive compounds, elucidate the structures using spectroscopic methods such as NMR, GC, mass spec. We also like to sequence the genomes, and we like to look for those biosynthetic clusters, those assortment of genes that will put together those molecules. And that could be representative right here with this biosynthetic cluster. We can use bioinformatics and actually take a really in-depth look at those genes, and can predict structures in a lot of cases, and even assign stereo centers. We can take those biosynthetic clusters and heterologically express these. Maybe we want to increase titers of a specific compound. Maybe we want to create derivatives that are otherwise inaccessible using traditional synthetic methods. We can do that using a variety of synthetic biology applications we have in the lab. We can also take that biosynthetic cluster and use that information. Maybe there's a new biochemical reaction that's never been seen before, and we can characterize the machinery that installs that certain modification and develop that as a biocatalyst, so that we can use that to maybe install that same decoration on another agent and maybe enhance its bioactivity, maybe decrease toxicity. We can also take this cluster and maybe we don't see its activation when we culture these strains. And so we can play around with certain regulators and try to turn on certain genes in hopes that maybe it is producing a bioactive compound. So we do use a lot of different techniques. So we are a multidisciplinary lab, which is fun because every day is different. And when we kind of apply these different approaches to antimicrobial discovery, one can think of as that, what do we do with antimicrobial discovery? And what are we really targeting? And if we look here, we can see a list of some of these bacterial and even fungal threats that the CDC is really kind of, emphasizing, because we don't have antibiotic agents to target antimicrobial resistance that we are observing with these pathogens. And so again, these are some of these CDC, like most, this threat list that we have, and some of the associated diseases that these pathogens do cause. And so a lot of these we'll also see are gram-negative organisms. And so when we're looking at developing new antimicrobial or even antifungal agents, again, turn to natural products. And I think this is a beautiful timeline that was published quite a few years ago that really demonstrates, the two methods of antimicrobial discovery. We can look at chemical weaponry found in nature, and those agents, these natural products, are highlighted in red. Or we can look at synthetic agents that have antimicrobial properties, and those are highlighted in black. And we can see that the natural products are really kind of outnumber the synthetic compounds here. And we can also see that this holds true when we're looking at antifungal agents. I'm going to highlight a review that was published last year by a grad student in the lab, Billy Hurd. And we can also see it the same thing with antifungal agents, where if we're looking at antifungal agents used to treat systemic fungal infections, two of the three major classes, so the echinocandins and polyenes, are natural products, where the azoles, shown down below, a huge class of antifungal agents, are going to be the synthetic compounds. So if we're looking at, looking for new antimicrobial agents for antifungal, antibacterial, even antiviral, we can turn back to nature. And so we can think of a lot of these agents that we have today that are clinically used came predominantly from terrestrial or soil derived organisms, bacteria and fungi. And if we want to find new antibiotic agents with new modes of action, where do we look? And we can think of is that environmental pressures can influence the chemical diversity of natural products, and microorganisms found in extreme environments may produce natural products not observed in their terrestrial, marine, freshwater counterparts. And so these organisms will serve as ideal resources for the identification of new natural products, potentially with new scaffolds, new modes of action, and furthermore, for the identification or characterization of new biochemical reactions. So this is where we're going to turn now to Great Salt Lake. And so Great Salt Lake is a hypersaline lake located in the state of Utah. It is a shallow but large lake. It is also a terminal lake. So that means that water entering the lake only leaves the lake through evaporation. And so salts and minerals are retained in the lake and concentrated over time. This is why the lake is a hypersaline lake. The south arm, shown down here, usually will have most of the inflow with fresh water. And this is in the springtime when the snowpack is melting. But because a causeway was built in the late 1950s, the lake is actually divided into a north arm and south arm. And the north arm, unfortunately, does not have much freshwater inflow. And this is why it's about 10 times the salinity of the ocean, whereas the south arm is about 6 times salinity. But because that causeway was built, it prevented the normal mixing of the water. And so Great Salt Lake, even though it's itself an extreme environment and a very unique environment, it is divided into 2 sub-ecosystems because of that railroad causeway. Because prevents the mixing of the waters, you're also going to have different microbiota between the north arm and the south arm. And that's beautifully shown here in this photograph taken from the International Space Station in the summer of 2019, where the north arm is this pink color due to the presence of carotenoids that are produced by archaea, predominantly found in the north arm, whereas the south arm is inhabited by blue-green algae. So this is due to presence of chlorophyll. And when we start looking at the different microbiota, and this was beautifully highlighted by Jamie and by Abby, we know that the microorganisms in the lake are really the foundation of the lake and this fragile ecosystem that is disappearing at a dramatic rate. And because of the hypersaline environment, not a lot of organisms can really survive in Great Salt Lake. We have a lot of brine shrimp and brine flies that feed on the microorganisms dwelling in the sediment as well as in the water column. And these brine shrimp and brine flies are crucial for the lake because they serve as food sources for millions of birds that migrate through North America and use Great Salt Lake to rest, to feed, and to nest. And so if you start looking at kind of the whole hierarchy of Great Salt Lake, we can see that microorganisms form the foundation of that food web, and that they're also important for a lot of these biochemical processes. So the recycling of nutrients for photosynthesis. And so again, it's just such a fragile ecosystem, and we really need to preserve everything in the lake. And what we're doing is also looking at natural product potential of these microorganisms and looking to see what types of specialized compounds they're making. We know that they're responsible for a lot of these biochemical processes for the health of the lake. And we also want to see what types of specialized compounds are they making that we could say, selfishly for us. And we started looking at the literature, for what types of compounds, are found from microorganisms in Great Salt Lake. We really couldn't find much. And so we wanted to think of, okay, Great Salt Lake is such a unique environment. These microorganisms should be very unique because, where else does this ecosystem exist in the world? And so if we kind of think again, going back to what's the natural product potential? How unique are these compounds, if they are making any, can we find in Great Salt Lake? And so because we really couldn't find anything in the literature, we thought, well, kind of think of low-hanging fruit. These are what we'd call like watermelons and pumpkins. And so let's really explore these organisms. And so we took our first isolation trip in 2017 to the south arm of Great Salt Lake, which is great because it's about a 30-minute drive from the University of Utah. And because not a lot was known from a natural product perspective, we really wanted to get the most bang for our buck. And what we wanted to do was really isolate actinobacteria. We know that actinobacteria are prolific producers of natural products. And if we could identify and isolate actinobacteria from sediment samples, we could culture these in the lab and really analyze their natural product potential and compare the structures to databases that are well established to organisms from the marine environment, from the terrestrial environment, from freshwater, to really see how unique are these organisms, and more importantly, how unique is the chemistry that they are producing. And so we desiccate the samples, which doesn't take much given how dry it is here, but we do desiccate in order to enrich for these spore-forming actinomycetes. We then plate that sediment, much like you would put sprinkles on a cupcake. We add that to agar that we make with Great Salt Lake water. And if you can draw your attention to these chalky organisms here on the plates, those are the first actinos that we isolated from Great Salt Lake sediment. We then took these organisms and we sequenced them by 16S and found that they were most closely related to marine-derived streptomyces as well as marine-derived nocardia. However, we were really trying to identify how unique are these organisms so that we could then really analyze their natural product potential. And even though 16S is commonly used to identify bacterial isolates, some complications can arise. You can get misdesignations due to partial 16S sequences, and that's what we were using to create this phylogenetic tree. You can also get ambiguous sequences. This is just due to conserved sequences across genre. So again, we really wanted to see how unique these bacteria are in Great Salt Lake and then relate that to their natural product potential. So we sequenced the genomes of our first 10 actinomycetes that we had isolated, and we turned to a new platform developed by Nadine Zimrit's lab at the University of Tübingen. This is called the Automated Multi-Locus Species Tree, and this is free software. So if you have genomic information or genomic data from your favorite bacterial strains, it doesn't have to be fully assembled. You can put in scaffolds or contigs. You put it into this platform. And what this MLST platform does is it makes high resolution trees de novo. And how it does this is it scans your genome sequences and it looks for conserved genes that are unlikely to be horizontally transferred, as well as single copy ubiquitous housekeeping genes. And so it uses these genes to create high resolution trees. And based on the genomes that you put in, it also puts in type strains as well as outgroup strains. And so the first tree I'm going to show is with our streptomyces, and I don't expect anyone to be able to read anything, but I've highlighted where our streptomyces, where they are in this tree. And if we zoom in, we can see that our streptomyces identified from Great Salt Lake sediment are more closely related to each other than they are to other streptomyces isolated from other hypersaline environments, as well as the marine environments. And so what this is representing, or at least indicating to us, is that these Great Salt Lake strains represent new phylotypes or new lineages. And we also see the same thing with our nocardia, where they're more closely related to each other, as well as to a hypersaline strain or nocardia strain isolated from a salt flat in chelate. So we have the genomes for our organisms, and really evidence is pointing that these are new lineages. And again, we kind of go back to, we have the blueprints, right? We have the genome sequences for our strains. Let's take a look. We have a glimpse of biosynthetic capability, the different biosynthetic clusters that these organisms have. Let's see what their capabilities could be. And so using bioinformatic analysis, we use a variety of programs. So we can use automated programs such as PRISM and AntiSmash, which are commonly used in the natural products community. We also use our own internal command lines to search for natural product clusters. And what I'm showing here are the 10 genomes of our Great Salt Lake Actinomycetes, which with these biosynthetic clusters around the respective genomes. And if we zoom in on these biosynthetic clusters, we can see that all 10 strains have a wealth of biosynthetic clusters, which means they should be producing quite a few natural products. We can take these biosynthetic clusters and actually break them down into natural product classes. So what types of natural products are they going to be making? And so this is a biosynthetic gene cluster heat map that was constructed by Eli Brain-Horvath, who is a graduate student in the lab. He created a Python code to make this graph, or this chart, excuse me. And what this is doing here to kind of walk you through this is on the left-hand side, we have different natural product biosynthetic clusters. These are making different classes of compounds. On the bottom are some of our Great Salt Lake strains, so our Nocardia, our Streptomyces, as well as a marine Streptomyces strain that we have. And in the middle, we're just going to have different heat maps based on the number of biosynthetic clusters that each of these strains have. At the top, we have the total biosynthetic clusters for each representative strain. So we have between 25 to 35 biosynthetic clusters per organism. And then we can even break that down further into the total cluster types. And this is great because we can now start comparing what types of natural product classes do Great Salt Lake strains have compared to marine, compared to terrestrial, compared to freshwater. Maybe we can start classifying these as sort of, enhancing fitness to these unique ecosystems. And again, this is just showing them the number of those representative clusters. So this is just the heat map, and then this is just the actual numbers. And so we start looking at the natural product potential of our Great Salt Lake strains. We see that one, they have a wealth of clusters. So they should be making a number of natural products. But we also see that these clusters are unique. When we look at databases, we find not a lot of similarities. So this again is an indication that these organisms should be making very unique compounds. And so we just jumped right in. Let's just start culturing these strains. And so we do a variety of approaches in the lab. We use bioactivity guided isolation of natural products. We use bioinformatic guided isolation of natural products. And we also use let's just look to see what these things are making and look at UV profiles as well as unique mass spec signatures. And so the first strain that I'm going to talk about is Streptomyces species GSL6b. And this project was spearheaded by a postdoc in the lab, Dr. Gungwei Wu. And when he cultured GSL6b, he found the presence of high molecular weight compounds with an isotopic distribution indicative of dichlorinated species. And normally with natural products, we will see that a lot of halogenated compounds usually will have bioactivity, and that bioactivity is usually attributed to the presence of that halogen atom. And so we got really excited when we didn't find any hits to any databases with compounds containing similar molecular weights. And so Gungwei was able to isolate a variety of different heptapeptides shown here. And these are the first, at least the best of our knowledge, natural products reported from a Great Salt Lake organism. And so we named them the Bonnevillamides after the prehistoric Lake Bonneville. So these compounds are non-ribosomal peptides. And these compounds the structures were elucidated by mass spec, high-res mass spec, as well as 1D and 2D NMR, and the protogenic amino acids, their absolute configuration were confirmed by advanced Marphys. And what I really want to highlight in here that's kind of color-coded is that these compounds contained a variety of non-protogenic amino acids. And so all three of these linear heptapeptides contain these dichlorohydroxyphenyl, 2-methoxypropanoic acid. They all contain highly modified proline residues. They all contain this N-hydroxyvaline motif. And Bonnevillamide A actually contains this unprecedented 4-methyl-azetidine carboxylic acid methyl ester moiety. And at the time, the only natural product to contain this motif was Bonnevillamide A. A couple of years later, Ralph Mueller's group identified bioprolide A and C from the cystobacteria. This is myxobacteria. And then late last year, Dongchun Oh's group at Seoul University identified Bonnevillamide D and E from a Streptomyces strain that they had isolated from the exoskeleton of a carrion beetle. And so the difference between Bonnevillamide D and E compared to A is that while Bonnevillamide D and E contain this 4-methyl-azidine carboxylic acid unit, it's not the methyl ester. Bonnevillamide E also contains this dehydrated threonine residue. So very similar in structure to Bonnevillamide A. But what was fantastic is that Dongchun and I were graduate students together at Scripps Institution of Oceanography, and we were able to collaborate on this project where we were able to use Dongchun's genome for his Streptomyces strain that made Bonnevillamides, other Bonnevillamides, D and E, and use that to help close our genome for Streptomyces GSL6b because we were having some difficulties because we had used Illumina sequencing. And so we were able to finally close the Biosynthetic gene cluster for Bonnevillamide A, and that's shown here. Again, those arrows represent open reading frames. They're color-coded based on putative biosynthetic function and are pointing in the direction of transcription. So we see these big purple genes or these big purple arrows that encode the non-ribosomal peptide synthetases, which stitch together these proteogenic and non-proteogenic amino acid building blocks. But more importantly, we have a slew of genes that are dedicated to making these non-proteogenic, these unprecedented building blocks that we find in Bonnevillamides AB, or the Bonnevillamides from the Great Salt Lake strains. And so using this information, we can start putting together biosynthetic process for how these building blocks are formed, such as this 4-methylazidine carboxylic acid methyl ester, which arises from a simple precursor, such as acetylmethionine. This is catalyzed by the methyltransferase BNVI, which cyclizes this cofactor into the azidine carboxylic acid unit, which then BNVC is a radical SAM, which installs the methyl group to form that non-proteogenic amino acid building block 4-methylazidine carboxylic acid. We then can see how that dichloryl hydroxyphenyl 2-methoxybutanoic acid moiety is formed, and this arises from the proteogenic amino acid tyrosine. And so a lot of modifications occur on this amino acid residue, such as chlorination, reduction, deamination, methylation, to form this substituent, also known as saccharochlorin B, which was identified late last year from Sanjeev Nam's group, from a Saccharomyces strain. So this is actually a natural product itself, and we see it as a building block in the Bonnevillamides. And then finally, we can take the simple proteogenic amino acid valine, and we can then take that and modify it as well to form the N-hydroxy substituent that then gets incorporated into the Bonnevillamides shown here, using these dedicated biosynthetic enzymes found in the biosynthetic gene cluster for the Bonnevillamides. So it's kind of a cool process where, in order to assemble these compounds, it has all the unique, machinery to make the building blocks to make these compounds. And so nature is pretty fantastic at doing this in very energetically favorable fashion. And so the Bonnevillamides were the first natural products we identified, and it really highlighted that that Great Salt Lake microorganisms are making this really unique natural products. And so the next project are the Salinipeptins, and this is in collaboration with Professor Bill Fennicle at Scripps Institution of Oceanography. And upon fermentation of Streptomyces species GSL6c, we were able to identify high molecular weight compounds. And so we were able to identify peptides with molecular weights ranging from 2,000 to 2,100 Daltons. And looking at accurate mass and the isotopic distribution of pseudomolecular ions, we could not find anything in the database with similar proteins. And so this indicated, again, we identified new natural products. By 1D and 2D NMR, we were able to identify all 22 amino acids and put together, fragmented pieces. However, we could not unequivocally figure out the order of the amino acid residues just due to overlapping NH signals in NMR, as well as the presence of proline residues. And so to elucidate the structures of the 4 salinipeptins, we integrated bioinformatics. So we turned back to the biosynthetic blueprints. So we looked for nature for help and used that as a guidance to solve the structures of these 4 peptides. And those are shown here. And so these are 22 amino acid containing peptides. And this is pretty complicated the way it's shown right now, so Salinipeptin A, B, C, and D. So to help, to kind of highlight some of the uniqueness, I'm going to simplify these structures with a cartoon where the amino acids are now represented with circles, and they're going to be color-coded based on whether any post-translational modifications have occurred. And so all 4 salinipeptins contain 22 amino acids. All 4 salinipeptins contain this aminovinylcysteine, modification on the C terminus. All 4 salinipeptins contain a highly functionalized N terminus. So salinipeptin A and D contain dimethylamine. Salinipeptin B contains this N-oxide moiety, and salinipeptin C contains this unprecedented dimethylamidazolidanone functional group. The difference between A and D is that one of the threonine residues in salinipeptin D has not yet been dehydrated to dehydrobutyrene as observed in salinipeptin A. So that's the only difference. But what was really remarkable about the salinipeptins is that out of the 22 amino acids, 9 are D, and this was confirmed using advanced Marphys. And so the salinipeptins belong to the ribosomally synthesized and post-translationally modified class of natural products. And they are part of the smaller subclass of ribosomally synthesized and post-translationally modified peptides called the Linaridins. And at the time when we isolated salinipeptin A, B, C, and D, there were only 3 representatives of this Linaridin class of RiPP natural products. So cepamycin, grisomycin, and Legionnaridin shown here. And while the amino acid core between salinipeptin A and cepamycin is almost identical, except for an alanine to valine swap, the main difference is that cepamycin is reported to contain all L amino acids, whereas salinipeptin contains, again, those 9 D amino acids. And so it was really remarkable that we were able to, again, find very unique natural products from Great Salt Lake organisms, and more than double this small subfamily of RiPP natural products that also possess antimicrobial activity. So cepamycin and salinipeptin A have gram-positive activity. And so when we started thinking again, like we have, these rare RiPP natural products, these Linaridin salinipeptins, how do we have any other natural product clusters that could also be making salinipeptin-like compounds? And if we turn back to, all of our biosynthetic clusters that we had from those original 10 Actinomycetes, we could look to see that 2 more strains possess the biosynthetic machinery to make the salinipeptins. And so upon culturing those strains, this is a project spearheaded by Abby Scott, who we saw at the beginning of the lecture series, and David Galgos, who was a postdoc at the time. They cultured the 2 additional Great Salt Lake strains, so GSL 9 and GSL 20, both are Streptomyces. Both had the biosynthetic machinery reported to produce these salinipeptin-like compounds. And upon fermentation, we saw the presence of high molecular weight compounds with similar molecular weights to salinipeptin A, B, and C. And what I'm showing here are the LCMS traces of our original producer, the salinipeptins, GSL 6C, as well as the 2 newly identified strains, 9 and 20, and the retention times of the different salinipeptins shown with A, B, D, and C, respectively. So to see, are these strains, these newly identified strains, also producing salinipeptins? What Abby and David have done is isolate the A derivative from the 2 new strains, and by NMR as well as advanced Marphys have shown that these are not really A. While the amino acid core is identical to salinipeptin A, again shown for reference, salinipeptin E and F by advanced Marphys contain a different assortment of L and D amino acids. So again, A, E, and F all have 22 amino acids, all have that highly functionalized N terminus, all have that C-terminal aminovinylcysteine moiety. But A has 9 D amino acids, E has 11, F has 5. So this is pretty remarkable that we can see similar compounds, but the different assortment of D and L amino acids. And one can think of is that the different configuration is going to make the structures slightly different when we think about 3D structures. So this can change how they interact with their molecular targets. And so with this information in hand, we have the biosynthetic machinery responsible for making the salinipeptins in all 3 of those strains. And so we can start comparing the different machinery, the different enzymes involved in some of these post-translational modifying reactions. More importantly, the epimerization reactions. Why are some strains making 9 D amino acids where some are making 11, some are making 5? And can we use that information to make more active salinipeptins, enhance their bioactivity against these gram-positive organisms? We can characterize these new catalytic reactions, these new epimerases, through these heterologous expression platforms, and we can increase the titer because in the natural host, they're produced at very, very low quantities where we have to actually culture liters and liters in order to isolate enough for structure elucidation and more importantly for downstream activity testing. So this is where Abby is right now, is creating these heterologous expression platforms for the characterization of new enzymes, as well as looking at producing these unnatural derivatives. And so where we are now with Great Salt Lake is, our initial isolation campaign really focused on the south arm, but with Jamie's help and Ani's help, we've gone all over the lake. And so over the summer, we went back to, Antelope Islands. We took out paddle boards and we're taking sediment samples. We used the sampling device called the Mud Snapper, which allows us to take sediment samples from various parts of the lake without having to kind of wade through the lake and disrupt, these microbiolites and, this fragile ecosystem. We can go out on paddle boards, just take sediment samples wherever we want. We can take it off the causeway. Abby went out to the causeway this summer, took samples along the north arm, which is shown here, and the south arm. We also went up to the north arm. Oh my gosh, absolutely fantastic experience. Thank you, Jamie and Bonnie, for inviting us. And so we're really expanding, our stretch of isolating organisms, looking at their natural product potential. And I'm going to end this seminar with really kind of emphasizing that a big problem in natural product isolation is that we think about all the thousands of molecules that have been isolated and elucidated to date, and they've really come from traditional fermentation studies. And a problem of natural products is really how do we isolate new scaffolds? And with post-genomic era, we can look at the genomes of these organisms and look at their true biosynthetic potential. And we know that what we're seeing in lab is not a reflection of what these organisms can actually make. And in reality, what we see in lab is only really represents about 10% of the total natural products that they should be making. And so how do we tap into this resource? And what are those clusters actually making? Because we can propose structures, but if you can't see them in lab, it really doesn't do you much good. And I really think that this is a beautiful, beautiful quote by Whitney Smith, who was a graduate student here at the University of Utah. And this is a quote taken from his master's thesis, where he really emphasizes that when we take organisms out of their natural context and put them in a laboratory setting, this is a synthetic setting, they're not used to this. We expect them to behave the same. So we've removed any kind of challenge. We remove their natural setting. And a lot of these biosynthetic clusters are going to be highly regulated, and maybe they need a certain trigger. Maybe if those clusters actually produce an antimicrobial agent, they're going to be tightly regulated until that trigger is present. Maybe it's a competing organism. And so that chemical weaponry that's being made by that biosynthetic gene cluster, you're not going to see it unless that threat or that trigger is present. And so we have something very simple in the lab. It's if you want, a new antimicrobial agent, challenge your organism with those threats, and maybe it'll upregulate or activate a biosynthetic cluster that makes a certain natural product aimed at that particular challenger. And so we did this with another Great Salt Lake strain, GSL15, and this was spearheaded by Dr. Gungwei Wu. And what we're able to show is that when we have Staph aureus by itself, our Great Salt Lake strain by itself, and when we challenge the two together, we see the production of a new compound. And Gungwei was able to isolate this compound and elucidate the structure. And this is a trialkyl substituted benzene natural product. It's a polyketide that we named BE52211D. We also have identified the biosynthetic cluster that makes this compound. This is a polyketide. So these red arrows encode polyketide synthases, which stitch together the carbon backbone. We've confirmed this biosynthetic cluster using knockouts as well as enrichment studies with C13 labeled precursors. And this, I'm going to end with a story that happened last summer, which was a summer project with Amma and Jace. Amma was an undergraduate who came to us from Smith College, and Jace was a high school student, is that we wanted to see where other Great Salt Lake strains capable of, producing a similar compound? Did they have something with similar regulation? And through genome mining, Amon Chase found another Great Salt Lake strain, GSL6A, that had a similar polyketide cluster, similar regulatory elements. And when they cultured the strain by itself, they did not see the presence of that polyketide product. But when they co-cultured it with Staph aureus, that challenger, they saw the production of that polyketide. They were able to isolate the compound and elucidate the structure. So we know that if we see these certain regulatory elements in our clusters, we can challenge them with Staphylococcus aureus, and hopefully we'll get upregulation of those respective compounds. And so we're using these regulators now to scan other pathogenic gene clusters from public databases in hopes of that maybe we can upregulate those respective clusters by challenging with Staph aureus. And so we're also looking at those regulators in more in depth, and to highlight this company here where you can engineer glassware fermentation apparatus. We're looking at those particular cues now of what turns on these specific clusters. And this is great for, challenge experiments where you have the organisms co-culturing together, but you can separate them into different sides of the flask using these membranes where they can share the same media, they can exchange compounds, but they don't physically touch. So we can do really intricate transcriptomic analyses of these organisms. And so in summary, our lab is very focused on natural product discovery and development using Great Salt Lake microorganisms. I've only talked about bacteria, but we're also looking at fungi. And we use a variety of approaches to really look at the chemical potential of these organisms, their biosynthetic potential, and even who are they and how are they interacting with each other. And so lots and lots of exciting things coming from the lab. And so hopefully I've been able to convince you too, is that Great Salt Lake is a treasure trove of unique natural products, unique biosynthetic clusters, unique biochemical reactions. And so it's just again, really, really fun to go out exploring the lake and looking at this such a remarkable ecosystem. And so with that, I'd like to thank the lab members, really highlighted the work done by Abby. Georgia is also working on Great Salt Lake strains. Eli is working on Great Salt Lake strains. Jace and Amma. Billy she comes out, everyone comes out and does Great Salt Lake work, as well as Pong Changshen just joined the lab. He's not pictured here. And Taka has also joined us over the summer. Also like to thank all of our collaborators at the University of Utah, as well as Dong Chen, Bill, Bonnie, and Jamie for just amazing collaborations, our funding, and thank you for your time. And I'm happy to answer any questions.
Jackie, thank you so much. I do have a question.
Yes.
So I've been, coming out with you for a couple of years now, year now or so. And I am not as smart as you or your lab. And I don't, I've taken like,
please, you're like the queen. We learned so much from you. Oh my God. So,
but I guess it's taken me a year and a half to fully grasp all of what you're doing, because a lot of it is so complex and, such in-depth science. And so, tell me if I have this right now. So bacteria have known methods and compounds that can be used to help humans, that can be used to help humans survive for whatever reason, kill antibiotic resistant bacteria. And so to find those, we didn't know about it at Great Salt Lake. So we went out to the lake, you and your lab went out to the lake and you sampled and you just collected a bunch of gunk. And then you dried it and then you cultured it on plates. And then after that, you looked at those cultures and you sequenced them and you used, a fancy statistics program and computer nerds to help you understand kind of what those compounds were that they were making. And then you identified those through a database, a known database of natural products. Is that right?
Yeah, we also we do use bioinformatics, or we also use biosynthetic logic as well to propose what the structure should be. I mean, it is very difficult to go from genes to product, especially with certain natural product classes. Some are definitely easier than others, but it's a way to kind of say, okay, is this something worth our effort to try to isolate?
Okay, so then you identify those things. And then how does it eventually go into, like, when I take a pill to treat my, to treat my bacterial infection that I got from when I fell in the lake, when we christened a boat and I got a really bad ear infection? Like, how does it go from that to me taking my pill?
Billions of dollars in about quite a few years. But normally what we would do first, and what we normally do, is that we'll first test for antimicrobial activity. So we'll do a battery of tests against bacterial pathogens, such as the escape pathogens. And if we see activity, we also do cytotoxicity activity to see, well, is this compound just toxic in general and just killing everything? Which isn't a bad thing necessarily. We can sometimes repurpose those compounds, maybe through like antibody drug conjugates, using those as payloads. But once we find that a compound is active and it's specific to bacteria and not toxic to us, then you would usually you have to have tangible yields in order to move it down for like PK studies. You'll want to do there's a lot of assays that have to be done to see, okay, what is its serum level? Is it orally available? Trying to develop it, can it be a pill in that regard? And then you have to go through a lot of clinical trials as well. And so to move that through the drug discovery pipeline takes years. And unfortunately, a lot of natural products fall out early on. And a lot of that has to do with yields, is that the producing strain will not produce enough compound to do all the assays that you need to safely evaluate these drugs. And sometimes you cannot produce enough using synthetic methods. And so it's just sort of, well, what do? You need material. And so that's why we try synthetic biology strategies for those situations.
Oh, my head is spinning.
Sorry.
No, it's really cool. I'm so glad to learn about this. We do have a question, and this is from Aaron Oren, and he always asks some of the best questions. So here we go. Okay, so if I understand it well, the salinipeptins are assembled on the ribosome. How are the D amino acids encoded? And are there special tRNAs that carry them? How are they incorporated in the growing peptide when the stereochemistry is wrong?
Yes, so fantastic question. So when the ribosome is kind of going along what we would call a precursor peptide that's encoded in the genome, it's just like normal peptide synthesis, and L amino acids are actually going to be incorporated. And then once that peptide is formed, the epimerase will actually go through and isomerize those different amino acids and convert them from L to D. So that's why we're really trying to characterize the different epimerases from the 3 different strains, because it almost kind of go from left to right and just kind of read down the peptide. And we want to know, why are certain amino acid residues changed and others aren't? What's sort of the recognition pattern of that? And so that is going to be exciting, because one could envision you could develop those epimerases as biocatalysts. There's some out there in other ribosomally synthesized and post-translationally modified peptides. So Jorn Peele's lab has quite a few. These are radical SAM mechanisms or radical SAM enzymes that can do this as well with the polyphenamides. And so it's not super uncommon, but it's just a fun biosynthetic puzzle of why certain amino acid residues are epimerized.
Okay, another question. These are all hard. How halophilic, halotolerant, salt requiring are your Great Salt Lake actinobacteria? Do you intend to describe some as novel species as based on 16S tRNA sequences and your multi-locus sequence analysis? They may be quite different from the known Streptomyces and Nocardia species.
Yes, so by full genome sequence, they are indeed very unique. And other labs have also looked at Great Salt Lake organisms in the water column and have also identified new taxa based on 16S. For looking at the halotolerance, some of our strains, in particular Nocardia, do require salt to grow. And we're also doing salinity tolerance testing with a lot of our strains now, especially with our recent trip to the North Arm. And we can plate these sediment samples on different plates containing increasing salt, and can find that different organisms actually will grow. So it's can be difficult to look at salt tolerance, to look at halophiles versus extremophiles, but this is something that, yes, we are actually doing.
Cool. Well, we've taken almost an hour. Does anybody have any other questions that you want to ask? We promise there's no dumb questions. And if anybody has ideas for Salty Science seminars, please reach out, let me know. You can email me at jbutler@westminstercollege.edu or throw them in the chat. I don't see more questions. So Dr. Jackie, so somebody wants to hear about organisms' adaptations for salty environments, from micro to macroorganisms. We can do that.
Yes, I can do the micro, you do the macro. So
yeah.
we also have, and this is a project that Georgia is working on, a grad student in the lab, is looking at osmolytes that are produced by Great Salt Lake organisms, as well as siderophores, because there's also quite a few, there's a lot of heavy metals in the sediment. And so we're looking at certain natural products, not from a therapeutic aspect, but more from an adaptive aspect that enhances the fitness of these strains, and looking for those unique compounds that it would allow them to survive in these very harsh conditions.
Interesting. Hey, do you think that Bonnevillamide should be the next official Utah State biosynthetic pathway? Should we try for that?
Yeah, awesome. I can just see that. Yeah.
Okay, Dr. Jackie, thank you so much for your expertise. I know Bonnie and I can't wait to get in the field with you, and I'm sure that you all will hear more from Dr. Jackie Winter and her really awesome lab. So thank you for coming. This will be recorded. I will send you a link with the recording that you can see, and we will see you later.
Thank you so much.